Okay, so now the first part of the topics, okay, we have done, okay, regarding the gene transfer and gene editing. So we have done that part already. Now we continue to these three important omics study in gene tech. So it's omics, basically it's a study. Okay, so we're going to study on what? We're going to study on the gene. So the study on the gene is known as genomics. Okay, genomics, it's a study of the gene. Okay, and we do know that from the gene we can transcribe the gene into mRNA. So means that we can study the expressions of a particular gene by looking into how whether mRNA is produced or not. Correct or not? Okay, so the process of or study this mRNA is termed as transcriptomics study, or transcriptomics analysis. So transcriptions, right, based on the word transcriptions, and from the translations of the mRNA, we get the polypeptides, right? From the gene to polypeptide, and the folding of the polypeptide into precise three-dimensional shape give us a protein, the enzyme. So the study on this protein, a mixture of protein, we want to analyze the protein, so we term it as proteomics analysis. So in this part 16.2, we are going to look at this, what we call the genomics, study of the gene, transcriptomic study of the mRNA, the expression, and the proteomic study on the protein. Okay, so these parts are a little bit different from what we have learned in 16.1, where we look at how we transfer the genes, how we do the editing of the gene using the CRISPR-Cas9 system. But today here, we are going to use certain tools to help us to study in terms of the genomics, transcriptomics, as well as the proteomics. Okay, so first we're going to look at these two processes here. Okay, these two process. The first thing we're going to look at this, what we call as a PCR, polymerase chain reactions. Okay, the first technique here we need to learn for today, which is the polymerase chain reactions, are called PCR. So if you have a watch, okay, the CSI before, then this term PCR always come out. Okay, basically we want to amplify, okay, what we did, we do, okay, why we need to use a PCR. Basically we want to clone and amplify the DNA. For example, in a crime scene, we may get a very minute amount, okay, limited amount or limited quantity of DNA. Then if we want to do so many analysis, how we're going to do it, we can actually subject this sample to the PCR to amplify the DNA. So basically PCR or polymerase chain reactions mimics our DNA replication, but our DNA replication take place inside our body, but PCR will take place inside a machine called thermocycler. So this is the first technique we're going to look at it in order to study the genomics and as well as transcriptomics analysis. Because we amplify DNA, we can actually still remember, we can use a reverse transcriptase to convert the mRNA in, or to use mRNA as a template and then make the cDNA. So as long as you have a DNA sample, you will be able to use this polymerase chain reaction to amplify the number of copies. Okay, so this is the technique we're going to learn for the first part. Okay, polymerase chain reactions. Now, second technique we're going to learn later, okay, will be, and second part of today's topics will be gel electrophoresis. Okay, gel electrophoresis. So in this case, you can have a gel. Electro means electric, phoresis means separation. So basically, your DNA, we can separate the DNA. Okay, so how we can separate DNA or analyze the DNA, we can use the second technique here called gel electrophoresis. And this gel electrophoresis also can be used for proteomics analysis. So we can use it to analyze the proteins as well as nucleic acids, and we can use it to distinguish between the alleles of a gene. So basically, how the allele we can separate. Okay, for example, we have HBA allele compared to HBS allele. One is a normal allele, one is the mutated one. So how we can use a gel electrophoresis to distinguish between these two alleles. Okay, and we're going to apply both technique, polymerase chain reaction and gel electrophoresis, in terms of the DNA testing for forensic medicines. Now, what's a forensic medicine? In this case, forensics, for example, suspect in criminal investigation, in parental testing, forensic medicines. For example, we're going to look at, okay, the dead body belongs to who? Okay, we use DNA testing to find out, okay, the identity of a dead body. So this, we have to use the both PCR as well as the gel electrophoresis to help us. Okay, and the last part will be the transcriptomics analysis, where we're going to use a chip, okay, a computer readable chip, which is called microarray, to analyze the genomes as well as to detecting the mRNA in the study of the gene expression. So the study of gene expression also termed as the transcriptomics. Okay, so we're going to start it with the first one, polymerase chain reactions. Okay, so first we need to know, okay, first part, why we use a polymerase chain reactions. The first statement actually tells us why we need polymerase chain reactions. Basically, as I say, that sometimes the DNA sample that you have is in a very, very minute concentration, minute, so means a quantity. If you want to do it for many analyses, it might be not possible. Are you clear? So not possible to do it unless we can amplify it. Okay, so this polymerase chain reaction is a common laboratory technique used to amplify the number of copies of particular regions of DNA. So it's slightly different from the DNA and replication in our body, where we replicate the entire length of the DNA. But for polymerase chain reactions, we can do a targeted region. Okay, I will show you how we can do this for targeted regions, where we're going to isolate the particular regions for the analysis. So sometimes we do not need to analyze the whole length of the DNA, but a particular segments of the DNA. So this PCR actually can help us. Okay, so before PCR, how we can do this? Before PCR, then we have to use a bacteria to help us. Still remember, we use a cloning vector. So the cloning vector helps us to clone a particular segment, the gene. Are you clear? But now we do have the PCR. So it means that we do not need to use a cloning vector anymore. We can subject our sample to this PCR technique. Okay, and the process completely in vitro. In vitro means outside of the body, inside a machine or inside a test tube. Okay, so why we do need this? Because we want, what is the objective? So objective here is to provide sufficient, we want to synthesize sufficient copies of target DNA so that we can do the analysis. Are you clear? For example, in molecular work, we want to have the DNA cloning, we want to have the medical diagnostic, we do want to have the forensic analysis. So the objective here, we want to have as many copies as possible. So it means that we need to have the sufficient copy. So how the process actually take place? Where? So the machine that we use here is a thermal cycler. Okay, it's a thermal cycler. It's the thermal cycler. So from the name, thermal means temperature, cycler means recycling. So what actually we do here is we have to set. Thermal cycler basically is a machine to change, to regulate the temperature. And because in the PCR technique involves 3 important steps which require different temperatures. Okay, so step 1, for example, we need to heat up to 95 degrees Celsius. Step 2, we need to reduce the temperature to 65, and then increase back to 72 degrees Celsius. So 3 steps in this case. So we need a machine. If you don't have a machine, let's say, for example, we don't have the budget to buy a machine, can we do the PCR? We still can do the PCR, but you have to do manually. It means that you have to prepare 3 water baths, and then you have your hand, and you have to use the what we call this, the tube, okay, the PCR tube. You dip it inside, okay, 95 degrees Celsius for 1 to 2 minutes, and then change. So you have to do manually for how many cycles? Normally about 30 cycles. Okay, it will take about 2 to 3 hours to complete the process. So thermocycler is a machine that regulates the temperature to make the temperature suitable for the reaction to take place. Okay, so now, in the PCR process, what we need to add into the buffer solution. So basically, we have a PCR tube. So the tube for the PCR is called PCR tube. It's a very, very small tube. Are you clear? It's a very, very small tube. Normally, we can put in roughly about 50, okay, microliter of the solution, 50 microliter. So it means that it's a very, very small tube, and the wall of the PCR tube is a plastic, but it's very, very thin. Okay, so then we're going to add in our mixture here. So what is inside the buffer here? So what we do have inside the buffer, we have these 4 items. The DNA templates that we want to amplify. Then we need to add in the primer. I will tell you why primer is needed. And DNA polymerase, or more specifically here, we are going to use a special DNA polymerase. We don't use a human DNA polymerase. I will tell you guys why, but we're going to use a DNA polymerase from a bacteria, which is called Thermus aquaticus. So the DNA polymerase we call as TAQ, T-A-Q. T stands for Thermus, AQ stands for aquaticus. It's a bacteria name, actually. It's a short form of bacteria name called Thermus aquaticus. So it's a TAQ polymerase. Okay, and then we do have the deoxynucleoside triphosphate, or it's called DNTP. In your ES syllabus, we do have this, but we call it as a free activated DNA nucleotide. Okay, so we're going to talk about this. So we have the ATCG. We add into the buffer. So with these 4 items and also the buffer solutions, then mix them well, we're going to start the process inside the thermal cycle. Okay, so we're going to look at one by one what is their functions. So first of all, we're going to look at the DNA templates. DNA template is a sequence of the bases of interest that we want to amplify, right? Okay, you don't simply put in any DNA, but it's a DNA template that you want to amplify. For example, we want to study on the gene. Okay, we want to study on this particular gene code for, let's say, blood group. Then we can actually use this DNA, use these segments, and then we put it in. Okay, and we do need the DNA primer. Okay, now, I'm not sure, I mean, in AS, do you guys learn about this primer or not, the RNA primer? But here we use a DNA primer. So the idea here, why we need to use a DNA primer, because of two things here. Okay, first, because of this DNA polymerase. DNA polymerase is a bit special. DNA polymerase cannot recognize single-stranded DNA. There must be double-stranded DNA. Are you clear? So it means that once you can, okay, this is a template DNA. Look at the diagram here. This is a template DNA. So this template DNA, when we separate it, right, semi-conservative, right, the process, so it means that with these 2 strands will act as a template. But if you look at this blue color strand, this is single-stranded. Can I see that? Single-stranded, single-stranded. So if a single-stranded like this, DNA polymerase won't be able to add the nucleotide one by one, that complementary to the template strand. Impossible. So in order for DNA polymerase to make a complementary strand, we need to add in a primer. So it's a set of primer. You have primer 1 and primer 2. So attached to a one end, so that now you can see that at these regions where I highlight here, it becomes double-stranded. Double-stranded. Can you see that? Double-stranded, double-stranded. So now DNA polymerase know where to bind and start the elongation process. Can you see that? Primer, attach, and then elongate. So elongate. So direction always from 5' to 3'. Are you clear? Okay, from 5' to 3'. Continue, continue. So we get 2 double-stranded DNA. Okay, and also primer will tell where DNA polymerase bind and initiate the elongation process. So if a single-stranded, then because we do know, right, for example, this is my DNA. I don't want to amplify the full length of my DNA. I just want a particular segment. So how we can do and tell the DNA polymerase, oh, this is the regions that I want to amplify, not the entire length. So primer can help us to bracket out a particular, okay, what we call these regions of the DNA that we can amplify later. Okay, and primer must be specific, okay, because it must be complementary. Can you see that? Roughly about 20 nucleotide long. We don't want to be too long. We don't want it to be too short. What will happen if too short? If the primer too short, you're going to have unspecific binding. Now, what is mean unspecific binding? If you look at these, guys, This is my primer, GATCAGGATACT, and the whole line you won't find anything complementary. Then it binds to a very specific region. Can you see that? But if let's say I do have a very short primer, let's say our primer here is TA. Then you can see that TA can bind here, too short. TA can have unspecific binding. TA can bind here, unspecific binding. Okay, so TA can bind here. So it means that it will bind at the unspecific region. So primer cannot be too short, roughly. Okay, write down roughly about 20 nucleotides long. Okay, the length roughly about 20 nucleotide long. Okay, it's a more, it's what we call this ideal one. But if too long, what will happen? If too long, then we may have the problems in the process called annealing process. May not take place too long. Are you clear? If too long, annealing process may not be able to take place. Okay, so a specific primer that are complementary to DNA target are selected beforehand, and most of the times we can custom made it in the laboratory. You can purchase it. Okay. Now, DNA polymerase. DNA polymerase here is the highlight. It must be a heat stable enzyme. That's why we cannot use a human. We don't use a human or any animal polymerase. Are you clear? Why? Because the entire process, just now I tell you that for PCR technique to take place, we need to heat up. First step, we need to heat up to 95 degrees Celsius in the mixture. Second step, we're going to reduce the temperature to 65 degrees Celsius. And third step, we're going to increase it to 72 degrees Celsius. So if you look at this temperature, so denaturations of DNA polymerase or human DNA polymerase confirmed. So that's why before the discovery of the tag polymerase, PCR technique is on paper only. We can do it, but without the tag polymerase, we won't be able to do it. Because why? Human enzyme won't be able to function or will be denatured in the PCR process, because we need to heat up to 95 degrees Celsius, followed by 65 and 72. All this temperature is too high. Definitely, it will going to denature the human's DNA polymerase until the discovery of this heat-resistant highlight. Guys, I forgot to highlight in this case, tag polymerase, TAQ, tag polymerase. So this tag polymerase is also special. We obtain it from the Thermus aquaticus. It's a bacteria that's commonly used. They're commonly found in these hot springs. Hot springs basically means that they live in the hot and high climates, high temperature climates. So it means that their enzyme will be the heat tolerant enzyme. Okay. So you can see that in this diagram just now, we have the primer 1 and primer 2 attached already. Then our tag polymerase now can recognize it. Tag polymerase now they can recognize the double-stranded and continue. Can I say double-stranded and continue to elongate? So add this nucleotide, but they don't add in simply any kind of nucleotides. They add in what we call the nucleosides or deoxynucleoside triphosphate, or we call DNTP. In our AI syllabus, we call it as a free activated DNA nucleotides. So what's the difference between the normal nucleotides and also the DNA, this DNTP? Okay, so this DNTP basically they have deoxyribose, so nothing similar as our DNA. Okay, nucleotides, nitrogen bases ATCG, but instead of one phosphate group, they have 3 phosphate group. So we're going to look at this Y3 phosphate groups. Okay, so sorry. Yeah. Now, let me show you normal nucleotides. The normal nucleotides will have deoxyribose as usual, then nitrogen base, and also 1 phosphate group. So this is a normal nucleotide compared to DNTP. Okay, So DNTP of free activated DNA nucleotides, they are going to have deoxyribose as usual, nitrogenous base, ATCG, one of them, but instead of one phosphate group, they have 3 phosphate group. Okay, they have 3 phosphate group. Now, what's so special? Why we need to have 3 phosphate group? Okay, triphosphate, triphosphate. So nucleoside triphosphate, D stand for deoxy because it's DNA. So why we do need this, what we call the DNTP in the process? Now, look at this. So we do know that if we have the primer and with this, okay? Okay, this is a primer end, okay? So primer short fragments, so let's say I draw another one. So primer, short fragment of primer. Okay, so this part we call as a primer. So do you realize that primer, the empty end here, this is a 5 carbon, so it's a 5 prime. Okay, 5 prime end. So if the anti-parallel system here, so this is the template. Anti-parallel, right? Okay, still remember? So if this is my template, okay, I draw another one. So this part is my single-stranded template. Okay, so it means that DNA polymerase now, DNA polymerase can recognize. Why? Because with the primer now, this part, can you see that? It's a double-stranded. Are you clear? Double-stranded. Okay, so my tech polymerase can recognize these double-stranded regions and start to add the DNTP. So look at the template strand. Template strand here, this is the 3 primes, anti-parallel. Here is 5 prime. Okay. Now the nucleotide add in here is a DNTP. Why? Because the process requires energy. Because when you synthesize, like for example, you synthesize glycogens, when you synthesize a macromolecule, we do need energy. Are you clear? We do need energy. Energy requiring process, anabolic reactions, right? Like protein synthesis require energy. So in this case, how energy is needed here is the nucleotide actually, first, the normal nucleotide will be phosphorylated. Normal nucleotide will be phosphorylated. So it means that ATP going to donate 2 phosphate group. So this become DNTP. So it's something like ATP. Can I see that? But ATP is a ribose, but this one DNTP is a deoxyribose. Okay, can I see that the sugar there, the deoxyribose? Now, what actually happened here is, so DNA polymerase or tech polymerase start to add the DNTP that complementary to the template. Okay, with triphosphate. So it add. So in order to form this sugar phosphate backbone, hydrolysis or removal of 2 phosphate group actually take place. So when hydrolysis is something like our ATP, right? ATP when hydrolyzed, remove 1 phosphate group, ATP become ADP, energy is released, right? So this energy is used to form the bond. Can you see that? Are you clear? Okay, so continue another one. Oh, sorry. Okay, so 3 phosphate group. Okay, add it, the complementary base. And then what happened here is the hydrolysis. The hydrolysis of 2 phosphate group. Can you see that? The hydrolysis of 2 phosphate group now release sufficient energy for the formations of the phosphodiester bond. And you can see that the elongation actually take place from the 5 prime end all the way to 3 primes due to these reactions. Okay, so DNTP basically they have additional 2 phosphate group, and the hydrolysis of these 2 phosphate group provides the energy or supply. Don't say produce energy. Energy cannot be produced, but supplied or provides the energy to form this phosphodiester bonds. And you can see that now the chain elongation from 2 just now become 4. So elongations with the directions always 5 prime. Here is 3 primes. So direction 5 prime to 3 primes. So again, this is the DNA replication that take place outside our body. Okay, take place outside our body, or we call it in vitro in the PCR technique, semi-conservatively. Can you see that? The process is still semi-conservatively. Okay, so come back to this, our notes. Now understand why we use the DNTP or free activated DNA nucleotides. Okay, so we need to add 4 of them. So this one's A, G, C, T. Can you see that? A, G, C, T. So just now we used the word DNTP, the N, oops, a mistake. DNTP, the N actually represent A, T, C, G. So we add 4 of them inside, okay? So the principle of this PCR actually is very, very simple because we just need to change the temperature. So in our syllabus, you can see that we're going to involve the denaturations, annealings, and elongation process. So what's been denature? So first, look at this. First step, we have the DNA template. Okay, so carry out. We heat it up to 95 degrees Celsius, where denaturations take place. So what's been denaturation? We denature the DNA. Because DNA is double-stranded, there are many, many hydrogen bonds formed between the complementary base pair. So using the heat energy, heat up to 95 degrees Celsius, use the heat energy to break the hydrogen bond. So it means that the function is something like helicase, but we don't use helicase in this process, but we use the heat energy. Okay, so we're going to get double-stranded. It's double-stranded. 2 single-stranded. Okay, we're going to get 2 single-stranded DNA. Okay, so as I say that, DNA polymerase or tag polymerase cannot recognize single-stranded. So we need to add what we call the primer. So we add primer at 2N. So the adding of the primer requires temperature. So it means that we reduce the temperature to 65 degrees Celsius so that annealing takes place. Annealing basically means that the attachments of the primer. Okay, so primer attached at the particular specific, what we call the complementary base sequence, and then we heat up to 72 degrees Celsius. We continue with 72 degrees Celsius, where elongation takes place. So now you can see that this is my template. Then my primer, and then elongation process take place. So from 1 copy to 2 copies. Are you clear? So this is only 1 cycle. After 1 cycle, so we're going to repeat it for up to 30 cycles. Are you clear? So the total DNA that we're going to get, estimated total DNA, will equal to 2 to the power n. N is number of cycle. Are you clear? Number of cycles. So 30 cycles, you get millions of the copy for us to analyze this from 1 copy of the DNA. So, okay. So for example, you get some specimen, very rare specimen, you won't be able to analyze them, but doesn't matter. We can always use the PCR technique to actually amplify it, and then we can do the study. Okay. So this elongation, basically the tag polymerase, we're going to add the nucleotide 1 by 1. So why we choose 95 degrees Celsius? The temperature is high enough to break the hydrogen bonds. So denature the DNA, making DNA from double-stranded become single-stranded, 2 copies. So 1 double-stranded to become 2 single-stranded DNA. And then we reduce to 65 degrees Celsius. Why we choose 65 degrees Celsius? Now, in our syllabus, 65, but in actual case, we need to do certain calculation. Now, in our syllabus, we choose 65 degrees Celsius because first, there are 2 things. It's not high enough to break the hydrogen bond. You cannot maintain 95 degrees Celsius because hydrogen bond will be broken. So when annealing process takes place, during annealing process, hydrogen bonds need to be formed between the primer and also the templates, correct or not? So if you choose higher temperature, no annealing. Are you clear? No annealing process. If you choose too low the temperature, unspecific binding will take place. So that's why 65 degrees Celsius is quite optimum for the annealing process to take place. Are you clear? So why 65? Because for the annealing, so the hydrogen bonds can be formed between the primer and also the DNA templates. Are you clear? And then continue elongation. Why we choose 72 degrees Celsius? Because this is the optimum temperature for the enzyme tag polymerase. So it works the best at this 72 degrees Celsius. So that's why thermocycler actually help us to change the temperature up and down, increase to 95 degrees Celsius, break the hydrogen bonds, and then reduce it to 65 degrees Celsius so that primer can anneal, and then increase to 72 degrees Celsius so that tag polymerase now can elongate and complete the DNA elongations process. Okay, so with this, I have done for the theory part. I'm going to show you guys a demo to you guys how we can actually cut out. Okay, previously we used a registration enzyme to cut out, right? We use a registration enzyme to cut out a particular segments of the DNA, but now how we can use the PCR to cut out that particular segments of the DNA. Okay, so pause the recording first. Okay, so now can you see that now from here I have these templates. I have this templates here. Okay, the one that highlights the templates, but I do not want to amplify the entire length. What I want to focus is somewhere here. This is the part that I want to focus, okay, where I'm going to synthesize many copy of this. So you can see that after 3 cycles, this is 0 cycle, means the initial. So now what happened here is I'm going to mimic the actual process take place. So now I put them into a thermocycler. So in the thermocycler, we heat up to 95 degrees Celsius. So when I heat up to 95 degrees Celsius, you can see that denaturations take place. Okay, denaturation process take place. Now, as I said that I don't want to copy out entire thing. I don't want to copy out entire thing. I just want to copy this segment. So I can attach the primer, but I don't want to attach a primer here. If I attach primer here, I'm going to get a full length, but I'm going to attach my primer. So I synthesize the primer. Let's say this is my primer. Okay, I use a green color to show my primer. So here I have another set of primer. Now, by right is 20 lah, okay, by right is about 20, but here I just use it. Okay, now because of complementary or anti-parallel system, if here is 3 prime, so means that primer will start with a 5 prime. You realize that my primer here is 3 prime, right? I highlight 3 primes, so my primer start with 5 prime. Okay, so this process requires temperature. We need to reduce the temperature. We need to reduce the temperature down to 65 degrees Celsius, allowing the primer to bind. If temperature is too high, the binding won't take place because the heat energy break the hydrogen bond. Okay, so what actually happened next? Our tag polymerase is going to add. Can you see that? It make a complementary strand. Same goes to the opposite strands now. So from 1 copy, now I get 2 copies. Okay, so this is after first cycle. So after first cycle, 2 to the power of 1, you get 2 copies now. You get 2 copies now. Okay, but I haven't cut it out yet. Okay, the 3rd cycle only you can see the effect. Okay, let me. Okay, so now I take these templates now. So again, denaturation process take place. Okay, denaturation process. So I have this is the primer. So we have to provide sufficient primer. Primer always start with the 5' end. Okay, primer always start with the 5' end. So again, now you can see that our DNA polymerase can recognize the double-stranded here. So when recognize, going to add, right, G, T, and so on. So you continue to add. Can you see that? Okay, and then here also the same thing, but now you realize that it becomes shorter already. So here will be 3'. Can you see that? This one become shorter, almost going to cut out already. This is after the second cycle. Can you see that? And this will act as template. This is only for the first molecule. So look at the second molecule. Okay, I'm going to fast forward now. Denaturation process take place. Okay, add in the primer. See that? And then add in the primer. And then tag polymerase is going to elongate. So tag polymerase elongate. So end with 3'. Can you see that? Here, this one becomes shorter already. This one becomes shorter. The rest still remain the same. But doesn't matter because we already almost get this what we call the templates here. And these 4 molecules, 2 to the power of 2, right? 4 molecules, right? So 2 to the power of 2, 4 molecules after the second cycle. Now, I'm not going to show you everything, but look at the third cycle when I use this. This one I don't need to show anymore, but I'm going to show you guys this. Okay, so denaturation process at the third cycle. Okay, primer add here. Can I see that? Primer when you add here. Okay, annealing process. So DNA polymerase is going to add 3'. And then here, 3'. So if you compare original length and now the length here. So this one later will become our template and continue to be used, and you can provide, many copy of our gene of interest that we want to cut out, or we want to insert it into the plasmid for the gene cloning, as well as for the gene expressions, right? Expression vector, for example, with the promoter, we will be able to express the product. Can you see that? Okay, so 3. This is cycle number 3. Okay, 2 to the power of 3. So in terms of calculations, you should get how many copies? 8 copies. From 1, you get 2. Then you continue, then you get 8 copies. And this will act as a template again for the 4th cycle. Can I see that? Okay, so I repeat. Now you can see that denaturation process at 95 degrees Celsius, and then continue with the annealing at 65 degrees Celsius. And then tag polymerase can recognize here, right, the double-stranded, and continue to add. Can you see that? Get more and more copy now. So this is the 4th cycle from 1 strand. Can you see that? So it means that after 30 cycle, you'll get 2 to the power of 30 molecules of sample that you can use it for the analysis. Okay, so with this, I have done the part here. So I stop sharing.